All right. So we're going to pick back up with the blood today. So last time we had talked a little bit about whole blood, the components of the blood, and started to dive into one of our specific blood cell types, our red blood cells or our erythrocytes. I think I got our questions working again. So when we were thinking about our whole blood and its parts, let's remember which part made up our largest portion of the blood first. Okay, let's take a peek. Great. Yeah. So if we're talking about our whole blood, we're talking about a big percentage of that as being fluid. Our plasma, it's got stuff dissolved in it. It's got proteins, it's got electrolytes. It's about 55% of your blood. Okay. So if you donate plasma, that's what they're taking, right? And our next biggest chunk are going to be our red blood cells. So that's about like 45% of the blood. If you measure the hematocrit is a number you might get back in test results. That's telling you about the volume of the red blood cells in your little vial of blood. And then our white blood cells and our platelets are super important, but they're only like 1% less than 1% of your whole blood volume. Okay. And we had seen this chart. So today we will at some point talk about our white blood cells as well, but just a kind of reminder, we're thinking about ultimately our red blood cells are erythrocytes. We know they're tiny. We know they're these little biconcave discs, so kind of frisbee donut things full of hemoglobin that lets us transport lots of extra oxygen around the body, as well as assist in carbon dioxide transport. So when we left off, we had been talking about how we make them, right? So we've got our hematopoietic stem cells in bone marrow. So they can become any kind of blood cell. That's kind of the point of this chart here, right? But under the control of certain signaling molecules, they're going to go down this pathway towards our red blood cell or erythrocyte. So you don't have to know this name, but they'd first become a myeloid stem cell. Then eventually they'd become something called a reticulocyte. So this is a number that you might get back in a test result as well as how many reticulocytes are there in the bloodstream. There shouldn't be any really. We should have our red blood cells losing their nuclei, losing their ribosomes, losing all their kind of cellular components to get small enough to become our mature erythrocyte, our mature red blood cell. But if your body is having to make new red blood cells extra super duper fast, some of those reticulocytes might sneak into your bloodstream anyway. So like if you're hemorrhaging or something. Okay. So mentioned that we need a signaling molecule to tell us to turn that stem cell into an erythrocyte specifically. So there are a bunch of different signaling molecules in your body. We're not really focusing on any of the rest of them, but you would be getting signaling to those hematopoietic stem cells telling you what type of blood cell to become. So that's what this first sentence means about cytokines called hematopoietic growth factors. That's our kind of general category for these signaling molecules that tell our blood cells what to become. We had one that specifically tells those stem cells to become an erythrocyte, to become a red blood cell. Okay. So we're looking for the name of our signaling molecule and what organ or organ system is making that signal. right, let's take a peek. All right. So we got the first part. We're looking for erythropoietin. When we're making a red blood cell, we're talking about erythropoiesis. Okay. The place that signaling molecule is going to be made is actually in the kidneys. And we'll mention that again when we get to our renal system and the kidneys. But the kidneys are all about sampling your blood and adjusting what's in it. So this is kind of why they have the job of telling your body it's time to make more red blood cells, because a byproduct of how they're filtering that blood and sampling it lets them kind of know when you don't have enough red blood cells, when you don't have enough oxygen. Okay, so this is where we left off, right? So we were making red blood cells, so synthesis of our erythrocytes, and we had left off on these dietary requirements. So we're going to need our iron for making hemoglobin, because that little heme part in each of those 4 subunits of hemoglobin needs iron. We got an iron atom in the middle. Okay, so you have to eat iron. You have to have iron in your diet. You can recycle some of the iron that is already in red blood cells that we're breaking apart, but that's not really enough to make all the hemoglobin that you're going to need for the like 200 billion red blood cells you're making every day. So we're going to need that. And we're also going to need folic acid and vitamin B12 in order to make more of really any type of cell, because these are necessary for DNA replication. But it's particularly noticeable when it comes to red blood cells because of how many of them you're making throughout the day. Right? It's like 200 billion a day, several million per second, right? So if you're having trouble with cell proliferation, if you're having trouble making new cells, right, you're going to see it in those red blood cells kind of first. This is a place that'll be really obvious. So folic acid and vitamin B12 for that cell division part. Okay. So that's how our red blood cells are born. We've got this signal coming from the kidneys, this erythropoietin telling us that we need to make new erythrocytes, which we're going to do in the bone marrow from those hematopoietic stem cells. We're forming all our hemoglobin while those cells still have things like ribosomes and DNA. But ultimately, losing kind of all that cellular machinery, all our organelles, losing our nucleus to become a mature red blood cell, a mature erythrocyte that's just packed with hemoglobin and not much else. Because we are packed with hemoglobin and don't have any of those organelles, all that cellular machinery, we know we can't just divide a mature red blood cell as it's going through your blood vessels. What we need to do is take the damaged old erythrocytes out of circulation, scrap them for parts, pull out the iron, and continue to make new ones in the bone marrow. So we're now going to focus on that removal part. Okay. So this is going to have kind of 2 pieces. So the filtering out of the erythrocytes in the first place is going to happen in your spleen. So your spleen is in your abdomen on the same side as your heart. So it's kind of on your left in the upper left quadrant. You'll see it later on. Okay. So it's going to be pulling those erythrocytes out of the blood. It's got lots of blood flow to it. And then the liver, on the other side of your body, so on the right, kind of just under your rib cage as well, is going to help out because it's going to help you metabolize those erythrocytes. So the liver is going to be doing that, pulling those red blood cells apart, helping us kind of recycle the iron that's in them. Okay, so how does this work? So in the spleen, we're going to have these leukocytes, actually, these white blood cells, the specific type called macrophages. And they're going to be helping us with this process because they're going to take our old or fragile red blood cells and they're going to kind of eat them. So phagocytosis is really kind of like enveloping another cell in order to break it apart. So our macrophages are going to do this as our red blood cells have been circulating through your bloodstream for a while and, I don't know, experiencing some damage as they go through things like narrow capillaries and stuff. Okay. So the macrophages in the spleen, pull out those red blood cells and start to break them down. And as we break them down, what we particularly want to think about is what's happening to the hemoglobin in those red blood cells, because they're basically little bags of hemoglobin. And we know that we need iron for hemoglobin, and you don't want to eat just like chunks of iron all day. So we're going to try to reuse that iron. Okay. So we're going to pull out our heme group out of our hemoglobin, and we're going to pull the iron out of the center of that heme group. That's going to leave us with some iron, and it's going to leave us with the rest of the heme, which is now going to be in the form of a molecule called bilirubin. Okay. So we're going to think about the iron in a second, but let's trace that bilirubin first. So the rest of that heme group minus the iron. So that's going to go back into your bloodstream, right? So we're sending that bilirubin into the bloodstream from the spleen, and it's going to travel to your liver to get broken down a bit more here. Okay. When we think about the liver in lab, we're going to see that the liver is really closely linked to our biliary system, our gallbladder, and they're both going to focus on producing and storing and releasing something called bile. So bile is going to help us a little bit in fat digestion when we think about digestion, but it's also going to be basically a waste product for us. So when we break down that bilirubin, we're going to send a bunch of the pieces that we don't need into the bile. They're going to go, some of them into the intestinal tract, and then some of them are just going to be in your blood for a while, back in the bloodstream, eventually make it to the kidneys, and then eventually get filtered out and excreted in your urine. Happens to be the case that the color of this bilirubin, the color of these molecules is yellow, which is kind of why urine has that yellow color. So this is a product of this breakdown. Okay. So that's what's happening to the parts of the hemoglobin that we kind of don't need to recycle. Now we're going to focus in on the iron piece. Okay. So we do want to keep the iron so that we don't have to constantly eat new dietary iron. We're still going to have to eat some, but this keeps the level lower, right? So we're going to be pulling that iron out, and we're going to move it around, and eventually we're going to want to move it to our bone marrow where we're making those new erythrocytes, those new red blood cells, because that's where they're going to have to make the new hemoglobin to put into those red blood cells. Okay. So we have two sort of molecules that go along with the iron. So we're going to have this compound transferrin, right? Somebody thought they were really clever here, right? Because you can see transfer in there. This is the form you're going to see it in when we're transferring our iron around the body. So when we're moving iron from the GI tract to the bone marrow, in the case of dietary iron, or from the liver to the bone marrow, in the case of this recycled iron, you're going to see this transferrin. The other reason this is clever is when you describe something as ferrous, that's a word for like iron containing. So I don't know, not quite a pun, but like a half pun in here, giving you both pieces. Okay, so that's that piece. The other way we may find our iron in the body is bound to something called ferritin, right? So you can see that FER root again, telling you that this has to do with your iron, okay? So when we're going to see iron bound to ferritin, that's going to be when we're keeping it for a little while in the liver or in the spleen or the small intestine before we need it somewhere else and move it. And we'll have a big diagram, kind of like all this stuff. So we're going to walk through this whole big picture from your book because seeing things in picture form can sometimes be helpful. Okay. So we're going to start out with the birth of our erythrocyte in the bone marrow. So here we see the bone marrow in the femur here, right? So you can see that femoral head, femoral neck going into the body of this long bone. Okay. And we can see where the bone marrow is here in the center of the bone. Okay. So we're getting the signal from the kidneys, we're getting that erythropoietin telling us to turn our stem cells into erythrocytes. This is where we'd be making the hemoglobin as well. When we're ready, that erythrocyte is born and goes into the bloodstream, comes out of the bone. Okay. It circulates around. It's carrying almost all of your oxygen throughout the body. It's also helping move our carbon dioxide ultimately out of the body. But after a relatively short period of time, it accumulates damage. It gets old. We're making like 3 million new red blood cells per second. So we can be done with this old red blood cell. Okay. So as it circulates through your body, all your blood eventually kind of makes it through all your organ systems, even though we're going to each organ in parallel. When you think about like any individual heartbeat, those red blood cells aren't just like directly assigned to one specific organ forever. So eventually that red blood cell is going to happen to go through the spleen. Okay. So when it gets through the spleen notices that this is not a very good red blood cell anymore. It's looking kind of fragile. We're going to break it up. So those spleen macrophages are breaking up that erythrocyte and its hemoglobin first into the heme part and the globin part. So we pull that kind of flower looking center of each of those subunits out, leaving the globin part, which is that protein polypeptide kind of coiled around. Yeah. And then we do a bunch of stuff. So we didn't mention what happens to the globin, but on this diagram, we can see what's going on there. So the globin part just gets pulled into its constituent amino acids. So pulling apart all those little beads in the chain. And that's also going to be a component of part of our bilirubin here. Okay. The heme group is what we're really focusing on because this is where our iron lives, right? So we're pulling the iron out of that heme group. We're going to send the other part again to the bilirubin, become this waste product that's going to go into the blood. And that iron has been pulled out in the spleen. We're going to be sending that around your body too. It's not going to live in the spleen for very long. So that is what we see here. So when you see this T-iron, that's telling you that it's transferrin bound to iron. And that's always how you're going to see it when it's in the bloodstream. Okay. So these are swimming along. And next we see what happens when they get to your liver, right? So first the bilirubin, which is basically the waste from our heme and our globin, goes into your bile. One place the bile goes is back into the bloodstream, ultimately ends up in urine. That yellow color of the bile is also why the plasma can be tinged yellow when you look at it. I think another arrow will pop on here at some point going down into the intestines from the liver for that bile too. But we're really focused on what's going on with the iron, right? So we can see here that potentially in the liver, we might store some iron. So here you can see on our liver, we've drawn F iron. So that's ferritin bound to iron, right? So it might hang out there for a bit. But since we're trying to get back to the beginning, to the birth of new red blood cells, and the creation of new hemoglobin, we can see that in the liver, we're going to notice that we can move that iron that's been stored as F iron, move it into the bloodstream, bind it to transferrin instead as the T iron here. Okay, yeah. Here come all the arrows in our intestines, right? So what's getting included here in this diagram is some of that dietary stuff, right? So say you cooked your dinner in a cast iron pan, that could be a source of some dietary iron, or you ate some kind of vegetable or food high in iron. So it goes through your mouth, through your esophagus, into your intestines, gets absorbed into the body. All right, so that's one way we can get another source of iron, right? So we're not just focusing on recycling iron. We're also getting iron from ingestion. once it gets absorbed into your body, it's in the bloodstream, again, it's going to be bound to transferrin, right? So it's T iron again, because it's moving around being transferred through your bloodstream. Yeah. Now, the other thing we see going on here in our GI tract is just a continuation of that getting rid of waste piece that's going on with our bilirubin and our bile. So we can see that some of the bile is getting excreted ultimately in feces. Okay. Our final piece is adding on our kidneys. So that's kind of the last system we need to think about, the last organ we want to think about as we're thinking about all the things that go on with our erythrocytes. All right, so since we're still on the death of our red blood cells, first we can see that that bile that has remained or come into the circulation and is hanging out in the plasma, kind of yellow in color. When it gets to a kidney, it's going to get filtered out and go into your urine. And as the kidneys are doing their job filtering stuff, they are also giving us the signal to make new erythrocytes. So that EPO, that's our erythropoietin, telling us to do more erythropoiesis because we've been killing off some red blood cells. So we got to replace them. We want to make new ones. Now, this is our whole circle of life for our red blood cells. Okay. Questions about any of these steps? Okay. I really like this picture. I don't know why. My recommendation for red blood cell life cycle is to just redraw this picture. Okay, but let's pull this in and think about some more kind of clinical stuff related to our red blood cell or erythrocyte life cycle. So one of the most common, most obvious kind of connections to this topic is anemia. So when we talk about anemia, what we're talking about is the fact that sometimes under some conditions, it's like harder for your body to carry around oxygen, right? So lower carrying capacity of oxygen. This is primarily going to have to do with those red blood cells and their hemoglobin. And there are a number of ways that we can kind of lower this oxygen carrying capacity, either through disease or by accident, or yeah, by circumstance, I guess. So the most common one is going to be dietary anemia, right? So the dietary is what I would kind of classify as like accidental anemia, right? So if you don't eat enough iron, right, then you don't have enough iron to make enough hemoglobin to put into your new red blood cells that you're making, right? So the result of this might be some like particularly small red blood cells because there's not that much hemoglobin packed into them. Not enough hemoglobin means you're not carrying around as much oxygen in your blood. Another kind of dietary anemia has its own particular name, right? It's got a special name, pernicious anemia, if you are lacking vitamin B12. So this is a vitamin that you should be getting through your diet or through supplements. That was one of the ones that was involved in the DNA replication and the cell proliferation piece, right? So if you have pernicious anemia, if you have this vitamin B12 deficiency, you might still have enough your iron, right? You still have enough iron to make enough hemoglobin, but you don't have the ability to make quite as many cells. So you might see something like extra large red blood cells that have more hemoglobin in them than usual, but there just aren't enough of these blood cells. So that's another way you can kind of damage the ability of your blood to carry around enough oxygen for you. So if you have anemia, you might be noticing like getting fatigued is one of the kind of signs of this, although often when you're fatigued, it's really hard to tell why you're tired, right? But this is one of the many potential causes. So these other forms of anemia likewise are going to damage your ability to carry around enough oxygen to your body. A little less fixable, I would say here. So hemolytic anemia can be caused by malaria, or sickle cell anemia is related to this too. So here we're looking at a red blood cell of someone who has sickle cell anemia. So sickle-celled red blood cells are shaped like that, like a sickle or a scythe, like the Grim Reaper kind of has, right? Slight change in the amino acids that make up that globin portion changes the kind of whole shape of your red blood cells. Now, this is a little protective, actually, against malaria, so it's more common in regions where malaria is prevalent. But you can see the cell is smaller. It's a different shape. It has more of a tendency to get kind of like stuck in capillaries. So we're lowering our carrying capacity for oxygen a little bit if we've got cells this shape. Aplastic anemia is a problem with the bone marrow, right? So you can also have too few red blood cells, too few erythrocytes, and therefore not enough oxygen because your bone marrow isn't making red blood cells as often as it should be. If your kidneys aren't telling your bone marrow that they should be making those erythrocytes, that's renal anemia. And then finally, we have hemorrhagic anemia. So this is, you don't have enough red blood cells and enough ability to carry oxygen because you're actively losing those red blood cells because you are hemorrhaging and you can't keep up with that. So all of these are kind of united together by this lowered ability to carry oxygen through the bloodstream, and pretty much all affect your amount of red blood cells or something about their size and shape. Okay. So that's what we've got on our red blood cells. And we're going to move next into our white blood cells, or our leukocytes, which is why I'm just pulling up this picture again real quick, right? So if we've got our signal erythropoietin, we become our erythrocytes, our red blood cells, but we've got other options here. So we're going to be thinking about our leukocytes and our platelets. So we can see that we've got a bunch of other types of blood cells that we're going to be walking through now. So not nearly as many of them as our blood cells, but they are really important to making sure that you don't get sick, that you can fight off infection, and that you can do things like recycle old bad red blood cells and keep some of those important components. Okay. So in general, our leukocytes, I think leuko means like pale or something, cytes means cells, so these are our white blood cells, are helping defend your body, okay? We consider them to be part of our immune system, right? So if you take a white blood cell count when you're looking at a blood sample, something that you're trying to figure out is this person fighting off some type of infection, some type of pathogen, right? Because you expect leukocytes to be made, particularly when they have work to do, although you're making some of them all the time. Okay. So they're going to be defending against different pathogens. So we're going to include like bacteria, viruses, parasites in that kind of category. They can also fight against cancer cells, any pathological cells in your body internally. And then they have the important job of basically being the cleanup crew for any old, dead, or injured cells in your body that don't happen to be cancer, right? But your body still doesn't want them just like building up and hanging around in your system. We want to get rid of anything that's not working. Okay. So if you were looking at a blood panel and you were trying to determine what's going on in someone's body, you might take a just white blood cell count, but you might also be interested in what type of blood cells specifically there are more of or fewer of. And our two big classifications here are based kind of on how they look. So we have granulocytes and we have agranulocytes. So when you see this A in front of something, basically means not. So our not granulocytes. And you can kind of see in the picture why we call them that, right? So our granulocytes, if you stain them and you're looking at them under a microscope to count them, you're going to notice they have these little granules, right? So these things that look like little pieces of sand here are the granules in the cytoplasm, hence cytoplasmic granules. Okay. So they stain different colors, potentially, depending on which type of cell you're looking at. But you can see that we've got them in these granulocytes. And in our agranulocytes, we can see nice, clear cytoplasm, none of these little condensations here. Okay. I listed them in a slightly different order than in your book because it helps me remember the granulocytes when I notice that they spell Ben, right? So this is Ben, my granulocyte family here. Okay, so we're going to have our basophils, our eosinophils, and our neutrophils with these cytoplasmic granules. And then we'll have our monocytes and lymphocytes in our agranulocytes. And some of our monocytes become macrophages. So those cells in the spleen that are helping break down our red blood cells, those spleen macrophages were once upon a time a monocyte. Okay. So we're going to go through our granulocytes first, and we're going to go among the granulocytes, we're going to go from the ones you have fewest of to the ones you have most of. So we have less than 1% of your white blood cells are your basophils. Okay. The important thing to know about your different types of white blood cells or your different types of leukocytes is whether or not they eat other cells. So eating other cells is phagocytosis. Basophils are non-phagocytic, meaning that's not their job. They don't do that. Okay. So not very many of them, less than 1%. They're not going to consume other cells, but they do potentially defend against large parasites. So that's one of potentially their jobs. So instead of engulfing a big parasite in your body, what they're going to do is try to release toxic substances outside of the basophil to try and kill it off externally, right? So we're thinking something that's probably too big for them to consume. However, hopefully you don't have too many large parasites in your body right now. So what we mostly notice basophils being involved in on a day-to-day basis is their contribution to allergic reactions. So our basophils release histamine and heparin. So when you have allergies to something and you're noticing like swelling and sniffling stuff, this is an immune response because your basophils think they need to defend against something and they're releasing these chemicals. So basophils, allergies. Next are eosinophils. Still not very many of them. Basophils were less than 1%. We got like 1 to 4% for our eosinophils. These ones could eat another cell. They are phagocytes, but that's not usually like the main thing they do. They are one of our types of granulocytes, right? And these granules have toxic properties in them, basically. So the toxic molecules in our eosinophils help you defend against some more specific parasitic invaders. The information on the basophils, to be fair, I haven't really checked it since your textbook was published, but you can see this may defend in your book's description. That's kind of vague because that's a little bit of a they don't totally know. But for the eosinophils, we do have more specific ideas of what they're defending against. So this would be things like parasitic worms. So they are sending out these toxic molecules to attack the parasites, usually. All right. Next, we've got our neutrophils. You have lots of neutrophils, right? So our neutrophils are like 50 to 80% of your white blood cells. So if you see something like an elevated white blood cell count, might be these neutrophils, right? Probably these neutrophils. They are phagocytes, so they are going to potentially engulf other cells in order to destroy them. They have their numbers increasing during infections, right? So they're pretty responsive to infection, which is why you might be counting them, why you might be looking for that elevated white blood cell count. And in terms of their life cycle or how they're operating, when they're first made, they're going to circulate in the bloodstream for a while, but not even really a full day, right? So just a couple hours, up to like 10-ish hours. Then they're going to move out of your bloodstream and go into your tissue, right? So they're going to try to chase down infections. They can do that because white blood cells are often actually able to get through pores of capillaries. So they can move from the bloodstream to tissue relatively easily. There's a process for this. Right. So those neutrophils that we're looking for elevation when we have infection are last of our granulocytes. So now we're going to think about our agranulocytes. Okay. So they're all going to be less common than the neutrophils because the neutrophils are up to 80% of your white blood cells, but they are super important too. Okay. So monocytes are going to be like 5% of your white blood cells. They can eat other cells, so they are also phagocytes. And like those neutrophils, they do only kind of circulate in the bloodstream for a couple hours. We don't have an exact time range we're pegging this to, but what we do want to know is that as they come out of your bloodstream, these monocytes, and move into tissue, they're going to become a new type of white blood cell called a macrophage. So you can see different in size and shape. So these are our macrophages that can potentially help us in the spleen process our red blood cells, break them down. We're going to see that some macrophages stick in the same place. Those are going to be fixed macrophages. So if you have a macrophage that's just hanging out in your spleen, right, that would be a fixed macrophage. But you also have macrophages that kind of move around your body, move through different types of tissues. So those are our wandering macrophages. So you'll have kind of both types, with some of them concentrated in specific areas of the body. Okay. Now, our final section here is a type of cell called a lymphocyte. Unfortunately, it has three kind of subtypes, so we will briefly mention those three subtypes in a second. But this is also a relatively big category here. So if you're doing a white blood cell count, most of that was probably your neutrophils, like up to 80%. But you might have a pretty high count of lymphocytes as well, so up to potentially 30%, right? If 30% of your white blood cells are lymphocytes, your neutrophils are not going to be 80%. That adds up to more than 100, which is why we had a range there on our neutrophils. They also can be found in the interstitial fluid. You mostly don't have a bunch of cells hanging out in your interstitial fluid, right? When we draw our diagram of the capillaries, we've got capillary interstitial fluid, then like body tissue, but sometimes you'll have some lymphocytes in there. Okay. So we're going to have our B lymphocytes, our T lymphocytes, and some null cells next. Okay. So for our B cells, or our B lymphocytes, their big job is to kind of mark pathogens. So when you're getting, say, a vaccine, right, part of what we're trying to do is train our immune system to recognize pathogens. So one of the things we would be doing is helping those lymphocytes recognize pathogens, right? So our B cells are associated with antibodies, all right? And they are going to help us mark other cells and tag them for destruction, basically. So when these invaders come into contact with our B cells, we produce antibodies when they contact those antigens in the plasma. Yep. Then we're going to see those antibodies, which are forms of immunoglobulins as well, moving around in the bloodstream. And they're going to kind of tell the rest of your immune system to get into gear. We got something we got to kill off. Okay. Another type, so type 2 of our lymphocytes are our T cells. So our B cells are just kind of like tagging pathogens, telling us what to do with them. Our T cells can directly actually damage foreign cells. So they can come into contact with any infected cells, but they might also come into contact with some mutated cells or transplanted cells, right? When we think about doing transplants, we have to be careful that we don't trigger someone's immune system to attack whatever organ we're transplanting, right? So the T cells would be involved in that. Okay. So when our T cells are maturing, they go through a process, but they develop into these cells called cytotoxic T cells that can then destroy their targets. So that takes a couple days, several days. They produce specific secretions. So kind of on the outside of the T cell, they produce these secretions that are going to form pores into the target cell to kind of bore into it and ultimately lyse that cell. So just like burst it, break it apart. Okay. So finally, our last type of lymphocyte is something called a null cell. Most of these are something called natural killer cells, which I think is the coolest of the white blood cell names. Their big job is to be your kind of early immune response to an infection, and they're particularly important when you're trying to fight off a virus. So they're going to attack cells that are infected with a virus, causing lysis, so causing them to burst, killing them off. And this happens pretty quickly. Okay. So we got all these different types of white blood cells, all these different types of leukocytes. Your book has a table breaking them down in kind of like a condensed form. I do recommend taking a look at that if you want it kind of all in one place. What we want to know about them in general, other than kind of like their main job and whether they're a granulocyte or an agranulocyte, is we want to know that they're developing kind of the same way the red blood cells were, right? So they're coming from those same hematopoietic stem cells, right? So heme saying blood, right? So can become a blood cell, stem cell. That's what that really says, right? So they're mostly going to be developing to full maturity in the bone marrow the same way the red blood cells were, although they get to keep like their nucleus and their organelles. Right? The exception to that are going to be our T cells or our T lymphocytes. To get to full maturity, a T lymphocyte actually has to go through your thymus gland. Thymus gland is a really important gland that's going to be pretty hard to impossible to see in an adult body, but it's really big in kids in the neck. In an adult, it just kind of looks like a glob of fat and it sinks down towards the heart. So unfortunately, probably not going to be able to show you a thymus gland in lab, but promise me they do exist, or I promise you they do exist. So the development of those cells is going to be controlled by various other kind of signaling pathways, these colony stimulating factors, and these molecules called interleukins. Okay. So you'll hopefully learn a lot more about pathogens and the immune response in some of your other courses. But that's what we got for our understanding of these cells for right here. We're just going to go through basics, kind of the names of what these cells are, and like a fact or two about what they do. All right. So we'll intro this last topic now. We certainly won't get through it today, but our final blood topic is thinking about hemostasis. So keeping your blood at a static amount, keeping your blood in your bloodstream, basically. Okay. So this is going to involve our platelets. So those little fragmentary little cell bits. So the last thing that we haven't talked about on that big flow chart of all our blood cell types. And it's also going to involve ultimately formation of a blood clot. So a larger clot that's going to be able to close over any openings in a blood vessel, so like any injuries. Before we get to our big blood clot, though, we're going to have some immediate reactions to an injury that act as kind of precursors. So we're going to see that we got a vascular spasm in response to an injury first. Then we form a platelet plug before we trigger the formation of a full blood clot. Okay. So we're starting out. You get stabbed, right? This damages your blood vessel, right? So this is the trigger for this whole process. We have this blood vessel damage. The first thing that's going to happen is that blood vessel is going to try to clamp down, right? So we had blood flowing through our blood vessel. It got cut. We want to minimize the blood loss immediately as fast as we can. So this vascular spasm makes this blood vessel smaller, right? Like this is a constriction. So this is vasoconstriction. So we now know how the effects flow, right? So when we make our blood vessel smaller, when we constrict, right, this is decreasing our radius, therefore increasing our resistance, therefore dropping down our blood flow, right? So this is starting to minimize our blood loss immediately by minimizing how much blood is coming to that vessel. This is an intrinsic vascular response. So this is just part of how they work, but it is also linked up to some sympathetic innervation. So that part of our autonomic nervous system. Okay. So we're going to start by constricting this blood vessel to hopefully stop so much blood coming to it. And then we're going to start setting up for our platelet plug to form. So we're going to see that the inner layer of the blood vessel that's been damaged, so that endothelial layer, is going to start to get a little sticky. Okay. It's going to start to get sticky because we've got these platelets running around in your bloodstream, and they're getting triggered to form something called a platelet plug. So if you want to name a platelet in a way that's kind of consistent with how we named our other blood cells, we might call them thrombocytes. So a thrombus is going to be a clot later on, but we can just call them platelets for our purpose. So they're going to try to form a platelet plug. So basically glue a bunch of platelets together at the site of the vessel damage to, in the relatively short term, block blood from leaving through this that injury. So this is going to decrease your blood loss. A platelet plug is not as good as a real full stable blood clot that we'll see later on, but it is like the necessary first step for eventually forming one. Okay. So our platelets, if you look at them, they're going to be colorless. They're teeny tiny. They're just cell fragments, no nucleus. Has a couple of organelles and granules still in it, though. So some things that our red blood cells didn't have. You've got a bunch of them in every milliliter of blood, so up to 500,000. And we'll talk about what they do next time.